Hi, online class. We just spent a couple minutes going over how to make a graph in Excel. I'll send you all a link to a video on how to do that. What we're looking at now is something like this. When we have an owlnet, we have an assignment, and we need to insert something. So we've typed our words, great. But we want to put an image in. We can do two different ways. And if you had a hard time with putting these in, this is the part where you pay attention. You can look and you can see this little picture icon. You can click on it. And then you can search for some image to insert, right? So you can search for, I don't know, you like this one right here, right? You'd insert some image, just like you'd attach an image almost to like an email, and that's going to put it in the body of the text. Or you can insert the link to something. So you have this here, insert link, and you again can search, and just like you'd attach a file on email. So this is the other option here. Boom. And then that puts it in there. Questions, comments? We're good? Great. On to photosynthesis, the exciting conclusion. It actually is kind of interesting. All right. So just to remind everybody where we're at, on Monday we saw this, right? We saw that light goes in and green light gets reflected, but most other lights get absorbed. Saw that. Anybody in this class already done their lab? Yeah, how was it? It was okay, right? Okay, good. Yay. I mean, I guess it's not a fair question to ask, because if you're like, I hated every moment of it, it's hard to say that, but okay. So what you're doing, you're seeing what pigments absorb different wavelengths of light. And you're doing a really, really shortened version of this. But if you were to do this a lot, you could plot here on, you could use Excel to plot this if you wanted to. You could plot down here on the x-axis wavelengths of light. And then so for a wavelength of like about 400, you could check chlorophyll A and see how much it absorbed that energy. So for 400 wavelengths of light, chlorophyll A absorbs a little bit. About 450, it's absorbing more. By the time you get to 500 nanometers, it's not absorbing like anything. But by the time you get to like 680 or so, it's back to absorbing some. And you can do that thing where you check a whole series of wavelengths for a whole series of pigments in something, and you can figure out where the plant is absorbing this energy of light. And you'll notice for like this standard plant, it does a good job of absorbing blues and purple light. It does a pretty good job of absorbing red and orange light. And green light right there is the kind that's not getting absorbed very well. Yeah? Just is. There are different pigments, right? So chlorophyll A and chlorophyll B, different versions of chlorophyll, they both work fairly similarly. Carotenoids are pigments that are shifted slightly over. Most of what carotenoids are doing are helping protect the cell from damage. They have a whole bunch of different pigments here. Great. So pigments here, beautiful. If you're shining different wavelengths of light on a plant and watching how photosynthesis is going, right? Like the rate of photosynthesis, is it going well, is it going poorly? You'll notice those rates of photosynthesis look a lot like where pigments can absorb light energy, right? So if you do something like this where you just shine just green light on this little plant, the plant's like, why are you doing this to me, right? But if you're shining shorter wavelengths of light, the plant's like, yeah, I'm cool with this. We're good? Sweet. And again, typically plants really like this whole, like regular normal light, right? Where you get the whole range. Okay. So that's all good and that's there. What we're at now is how this means anything. So what's going to happen is chlorophyll gets excited. It gets excited by light. And this is the picture. We have an electron that's in what's called ground state. It's not excited. It's low energy. It's just there. When a photon comes in and hits it, it excites it. The photon adds energy, bounces this electron up in energy. It's now excited, right? And as that electron drops back down in energy, it's going to give off energy. You see this in fluorescence, right? So it goes from low energy, boom, bounces up. It's excited state. As it goes back down to ground state, it's going to give off light and some heat. You can see this with anything glow in the dark, right? So you have your glow in the dark notebook or whatever. If it's not been charged, it doesn't glow. But you give it light energy, that light energy bounces those electrons up in energy. And then as they go back down to ground state, as they give off energy, they give off light. We're okay with this? Beautiful. That sets up the next part of things, which are the photosystems. So, lots of words here. Basically, a photosystem is this system that's responding to light. There are things called light harvesting complexes. So, there are pigments with some proteins around them. Personally, I feel like light harvesting complex sounds like something a supervillain would have, like, I'm going to go harvest the light, right? It's a good thing, though. Plants are doing this, they're harvesting the energy from light. It looks something like this. This is called a photosystem. The light comes in, drawn here as a lightning bolt. That photon has energy in it. Or is energy, depending on how you feel about this. That photon then gets absorbed by the pigment. That energy gets absorbed. The energy gets handed to that little green blob of a pigment, okay? So if you're thinking of energy as like a ball of glowing power, or I don't know what you want to think of it as, if you think of it as a blob of energy, that gets handed from one pigment molecule to the next, to the next, to the next. So it gets hot potatoed around, right? Energy up there, gets handed to the next pigment molecule, next. It moves down to here. Right there, where there's an electron, in ground state, just chilling. Not participating in a lot. That electron gets handed that blob of energy, and it goes from being a low energy electron that's just chilling to being a high energy electron, right? This thing is now very excited. That high energy electron then gets grabbed by what's called the primary electron acceptor. Everyone's with me so far? So, there are a couple different photosystems inside most plants. There are two of them, and they operate sequentially. So a photosystem does its thing, and then another photosystem does its thing. If you were left to your own devices, and you had two things, and this one happened first, and this one happened second, what would you call them? Like photosystem 1 and 2? Is that what you said? Okay, people like the idea, like photosystem 1 and 2, right? Makes sense intuitively? You're going to be disappointed. There are two photosystems. They're called photosystem 1 and 2. Photosystem 2 starts first. It goes photosystem 2 and then photosystem 1. And Mackenzie's feeling sad about this?
That's not how numbers work.
No, okay. So, but okay, yes. So that's not how you should do numbers. You're like, oh, okay. What happened was, when people were searching for this, they found what they thought was the photosystem, and they found it first, right? And then they find other photosystems, so they're like, photosystem 2. But that one happens first. So yeah, I'm sorry. But are we okay with this? Yeah, okay. So, but now you'll know forever, right? So two types of photosystem. Photosystem 2 happens first, and it's really good at absorbing wavelengths of light in that 680 nanometer range. And then photosystem 1 happens next, and it absorbs slightly longer wavelengths of light. What happens here? These photosystems harvest energy. There are two ways for them to do this: cyclical electron flow and linear electron flow. I'm going to show you linear electron flow. I have a take-home message at the end of this, I promise. I want you just to see it. So all these slides, so we're right here in the light reactions. We're not worrying about the Kelvin cycle right now. We're here at the light reactions. Light's coming in, water's involved, oxygen's being given off, we're building ATP, and we're making NADPH, all right? But the steps to get this done, they're here with green backgrounds. Don't memorize the green background ones, but I do want to talk you through it, okay? And I realize I'm like stuck here because I need the online people to know what's happening here. So, this is photosystem 2 we're looking at. Photosystem 2, some proteins, the pigments, and the little green blobs are right there. Photosystem 2. Light hits this thing, right? It excites the system. So now this energy gets passed along until it excites this thing. It's called P680. It sounds cool. When that happens, you bounce this electron up in energy. It used to be low energy. Now it's high energy. And that excited electron gets transferred to this primary acceptor, right? Phew. Which is great, right? So now we've got a really excited electron up there by the 2. Okay? Cool. That's great. This P680 thing, it now doesn't have its electron. It used to have an electron, but it just got bounced up, so now it's upset. P680, it's called P680 plus if it's lost its electron, is an oxidizing agent. It cares deeply about having electrons, like very, very deeply. It's the strongest biological oxidizing agent that we know of. Meaning, yeah, okay, the electron got excited, was given over to the primary electron acceptor, fine. P680 has to have an electron. We'll stop at nothing to have an electron. And it sees that there's water nearby. And it says, you know what? Tough beans bring water, and it breaks water apart. Well, it's water split by enzymes, but water split apart. Those electrons get transferred back there. So P680 plus goes back to P680, meaning now that thing's been recharged with a low energy electron. The electron in water is low energy. It got that electron there can hang out, low energy electron, ready for the next time that gets excited, then it'll go to high energy electron, you'll grab another low energy electron and keep refilling the system. So far so good? This is the part where you make oxygen, and it's kind of an accident. The plant's not there being like, you know what the animals and other life forms on Earth need? Oxygen. No, what it's doing is like, I need electrons, I've got water, I'm pulling electrons, hydrogen ions are hanging around out here, but I'm making molecular oxygen doing this. We're cool with that? Great. So far so good? Okay. So I'm one step off a little bit here. So the electrons are doing their thing over here. We now have up here, we have this really high energy electron. Really high energy electron. It's going to go down an electron transport chain. We've seen electron transport chains before, right? High energy electron, handed to next thing, to next thing, to next thing, dropping in energy. As it does that, it's doing the thing that you've seen before, where it pumps hydrogen ions out. And then things happen, and you make ATP. Okay? We're good? You've seen this before in mitochondria? This is now going down in the chloroplast. All right, and energy released by this is driving this ATP synthesis. So now we've got this at the electron went down, this electron transport chain drove the production of ATP. We have an electron sitting right here. Well, got an electron right here. Low energy electron. Additional light comes in, balances that electron back up in energy. And so a similar thing to what happened here. This is photosystem 1 now that we're looking at. You bounce this electron back up in energy, very similar to what we saw right here. Boom. It ends up being grabbed by NADP plus, and now it's NADPH. Again, we've seen this before, right? Where you grab a really high energy electron by NADP plus. We've seen a very similar thing before. So now we've got a high energy electron. We've got high energy electrons hanging out here. We're okay with that as a concept? Great. These high energy electrons are going to be moved over to the Calvin cycle to do stuff with. So the take-home message, it's this adorable picture. Can we all take a moment to appreciate the adorableness of it? It really is, right? Like it's like painfully cute. Okay. Right? I realize you're adults, but stay with me here. We have this low energy electron that's chilling here. Photon excites it. Phew. This is photosystem 2. As that excited electron goes down in energy, it drives the production of ATP. The electron ends up in photosystem 2. Another photon excites it again. The excited electron ends up in NADPH. Yeah? We've made some ATP doing this. We have this excited electron here. And we've made O2 from water as a byproduct here. All right? Everyone's feeling comfortable here? Got it? Okay. This is called linear electron flow because it looks like a line, right? You start here, by the end you've just gone straight through. There's another option. It's called cyclic electron flow, and it goes in a circle. This kind, you don't release oxygen. It looks like this. So light comes in, shoop, excites things. You excite this electron up here. The electron goes down an electron transport chain, driving production of ATP. And this now low energy electron gets picked back up, and you just can run the cycle again and again. It works, it's great, it builds ATP. It doesn't give you NADPH, and it doesn't give off oxygen. This kind, this is when, if an organism is only doing cyclic electron flow, they're only going to have photosystem 1, which again then feeds in a little bit to the like some bacteria do this, where they're going to run it cyclically. It's thought that this one in evolutionary time was figured out first, like, hey, let's, balance this electron up in energy, use it to drive ATP production. And then over evolutionary time, organisms are like, or we could get even more energy off, right? Oh, and this is purple sulfur bacteria, and it is legitimately purple. It's not been stained to be purple. It's purple. The stuff is weirdly purple. Okay. Everyone feeling comfortable with this? Linear electron flow, cyclic electron flow. Take-home messages. You've harvested energy from light, right? The NADPH stuff we'll worry about later, but this part right here, this part right here, the making ATP as you're transferring electrons down electron transport chain, we're going to look at that briefly. It looks really similar to mitochondria in many ways. Like you've seen this before, but backwards. So chloroplasts and mitochondria are both going to generate ATP. They both do that. The energy source for mitochondria is that chemical energy that you got from food. The chloroplasts are using light energy to drive this. And they're a little bit backwards. So here, you see this mitochondria right here? Outside, where there's just the white slide, that's the rest of the cell, okay? Rest of the cell, you've got mitochondria, it's got this membrane, it's got, now we're inside the mitochondria, then there's another membrane farther inside, right? Where it's darker gray, there's a ton of hydrogen ions. This is the intermembrane space. So during the electron transport chain, as high energy electrons are being passed along, the electron transport chain is pumping hydrogen ions, and it's pumping hydrogen ions from inside here out into the intermembrane space, right? And there's an ATP synthase right here. Those hydrogens that are hanging out here fall back in, drive the production of ATP. Cool with that? The picture looks the same, but flipped for chloroplasts. So out here we've got the rest of the cell on the white part of the slide. Then we've got the chloroplast right here. Inside here, these are the thylakoids, right? And it's inside the thylakoids, you have a bunch of hydrogen ions. So we've got this stuff right here. This is the stroma, the outside, well, the goo inside the chloroplast, and then the thylakoids in there. The electron transport chains are here on the thylakoids, and they're pumping hydrogen ions from the stroma into those thylakoids. And then those hydrogen ions are going to fall back out. ATP synthase, ATP synthase is going to synthesize ATP, right? We're okay with this? Same idea, but you'll note hydrogen ion concentration high here, high here. You see that? So far so good? Okay, doesn't blow anybody's mind? Great, so at the end of the light reactions, the one that took light, we have ATP has been produced and NADPH have been produced. The take home from this whole shebang is this slide. Is this too small? I have it bigger too. This is the whole thing. We're going to look at it bigger in just a sec. So, photosystem 2, which happens first, and it's, yeah. Light comes in, excites this pigment, it's absorbing the pigment, bounces electron up, the electron acceptor grabs that high energy electron. You need to replace the electron, so you steal electrons from water. You end up making some oxygen. You also make some hydrogen ions, because you broke the water apart, took the electrons, and left oxygen. So those hydrogen ions are now inside the thylakoid space. Super convenient. You want high hydrogen concentrations here. And oxygen leaves as a byproduct. Yay, oxygen. That hydrogen electron that you bounced up in energy, going down electron transport chain. As it does that, you're pumping more hydrogens in. Gets grabbed. We're going to stay with linear electron flow here. With that, gets grabbed over here by photosystem 2. Additional light bounces it up in energy again. that new high energy electron, it's put over here into NADPH, right? This NADPH heads for the Calvin cycle. The hydrogen ions that you've pumped in, or either made or pumped in, are going to drive ATP synthesis. And this ATP is also headed for the Calvin cycle. We're good? Excellent. Questions about that? All right, so we've harvested light. That's great. We've got the energy from light. Now we're on to using that energy to build stuff. So this is where we go into the Calvin cycle. Lots of things about the Calvin cycle. The take-home message is this. You're having carbon dioxide come in, right? Carbon dioxide coming in. You're producing sugars, you're producing organic molecules. Also, is this room freezing cold? Okay, I have no control over this, but it is, like, painfully cold. Anyway, sorry, just thinking about it. Sorry. Carry on. It's just people look stressed. Except for, like, one person who's like, yes, this is the only room I've ever been in that's actually appropriate temperature. Yeah. Okay, so carbon dioxide's coming in. Sugar's leaving. It leaves as, like, a little bitty sugar, but you can make it bigger. You're feeding the cycle energy from energy you got in the light reactions, from ATP and NADPH. As you run this cycle, you feed it this energy, you change these back. So you change ATP back to ADP because you used it. You change NADPH back to NADP plus because you used it. And this is our cycle. Like before with linear electron flow, don't memorize the green slides here, but I want to show you what's happening. You've got, by the end of this, we're making a little 3-carbon chunk. So we're going to need 3 carbon dioxides, because we need 3 carbons. Coming in, it's called carbon fixation. Carbon gets added to this little molecule here. It takes energy, ATP and NADPH, it takes energy to rearrange all the electrons and carbons where they're supposed to be. But after paying energy in, you can end up building this nice little 3-carbon sugar. Okay? That little 3-carbon sugar is called G3P. It's technically called glycerolaldehyde 3-phosphate, but like even its mother calls it G3P. Like it's just, that's because reasons. So that's going to leave. Then it's going to take some more energy just to get this thing back to where it was before. You need to put everything back so you can start the cycle again. We're okay with this conceptually, right? Carbon dioxide's coming in. You're spending some energy to rearrange everything. You're getting this G3P sugar out. You put everything back to be ready to go for the next cycle. Yeah? Okay. So, this is really, really good because it makes life sustainable. So, you might have noticed that the sun is great, right? Lots of energy there. Can you tell me about what happens at night? Read the sun? Right, like, okay, so the sun doesn't turn off, but from organism's perspective, it kind of does, right? Like, it had been there providing tons of energy, that was fantastic, and now it's gone, right? So tons of energy of the sun, beautiful, super important, but it's not constant. So, like, if you're just solar powered, the instant it's cloudy or there's not, or it's nighttime, you're done. That's a problem. And organisms don't just like instantly die as soon as there's no light, right? Hopefully not. So photosynthesis is the way to capture that light from the sun and put it in like battery form, right? So there's energy coming in, capture it in some form that you can use later. Because Because the energy's coming in, that's great, but you need to be able to hold on to it. And then later, when it's dark, when you're hungry, when it's cold, use it. Use that chemical energy, right? So photosynthesis is going to do that. It's going to get sunlight energy stored as chemical energy in organic compounds. So this is going to be great for like all life, right? So you can eat potatoes, you can eat apples, that kind of thing. It's going to make a bunch of the structure for plants themselves. It's also, as the byproduct, it's going to make O2, which again is really, really important for life as we know it, to have molecular oxygen to breathe. Questions, comments, concerns? Okay. I'm going to try, we're going to try something because it's helpful. I have 2 more slides to show you. And these are the slides, if you only have brain space today for 2 slides, these are the 2 slides. Like, you should know all the things, but if you only have brain space for 2 slides, the next 2 I'm going to show you. And then we'll come back to this. So, take home message, and I realize it's a little bit cheaty because this is a very full slide. But if you were to see this picture on a quiz or exam with like things blacked out and like what's given off here, you would know. You'd be like, that's oxygen given off there, right? This is the overview of the whole thing. And everybody knows that these slides are posted on Alnet, right? People know, okay, just make sure. So oxygen's coming into this plant, carbon dioxide's coming into this plant. The plant is giving off, sorry, did I say oxygen? Carbon dioxide's coming in, water's coming in, the plant's giving off oxygen and is giving off sugars. Inside, if you look at it close here, we have the light reactions. They're capturing that energy. Water is going to be torn apart. You're going to give off oxygen. These light reactions are going to give off oxygen, but mostly make ATP and NADPH. And those energy sources are going to feed into the Calvin cycle. The Calvin cycle's grabbing carbon dioxide, stuffing them together into nice little sugars, and using the energy stored here, these get replaced back to low energy where they can start another cycle. Beautiful. I really need to at some point bring in the Lion King Circle of Life video and just like, we all can listen to it briefly. Then this will be the summary of those 2 things. So light reactions happen at the thylakoid membranes, right? That's where you're capturing the energy. That's where you're splitting water, releasing oxygen. The Calvin cycle reactions, they're happening in the stroma. They're happening in the goo outside the thylakoid. That's where you're building that G3P sugar. And that's where you're returning ADP and NADP plus light reactions. Right? Thing just to remember, just for life, having chloroplasts doesn't make it so the plants can't have mitochondria. Plants have both. Plants have these chloroplasts. They're great. They do all these things. And then the plant is going to use the organic molecules it made to build stuff to a bunch of things, but it's also going to feed some of those into its own mitochondria and break them down. All right. Questions? Bear with me just a minute while I yap to online people. Online people, this things to know slide, go over it and with yourself or with a long-suffering roommate, try to see if you can answer it. All the rest of you, we're actually going to try. We've got 10 minutes. So make eye contact with somebody nearby you. Deep, intense eye contact. And ask them one of these questions, okay? If they don't know, and they try to cop out with like, I don't know, make more intense eye contact and say, try, or lie, or guess, or something, okay? I don't care if you guys are right or wrong. I do care if you struggle. Like, this is good. The struggle is good for you, okay? So they struggle through some answer. If they're completely off base, be like, no. And help them out. If they're great, great. Then, because you so rudely asked them a nosy question like this, they're going to ask you a question. These are good options. If you have some other question related to photosynthesis, go with that one. I'm going to like nosily pop around and, after I take attendance. So do your thing.
To make it look pretty, of course. Duh. Colors. Everybody likes colors. Oh, I have a question.
Yes, about photosynthesis and beautifulness. That is photosynthesis. Okay. I'm going to think too.
I was driving the other day, and it just popped in my head. So you know how it's fall, and so the leaves change colors, and then they fall down? But what if they survive the winter? Do they still fall eventually, or do they resurge before the it's all put in?
They're falling because the tree has that light. Briefly. So during summer, maybe it is, and again, different trees, right? But like the citrus trees, right? They're capturing light energy, great guns, and they're busy. Like, you know, berries go for like hibernation idea. They're busy craning themselves full of sugar, right? And then, in winter they're like, it's not working to try right now. Life's not just like flying off. So they basically stop supporting the leaf. They're like, good luck, and just like cut off the life support to it. Oh, the leaf dies and flops off. So if the leaf didn't die and flop off, yeah, it'd go like you could if you got super enthusiastic, you could keep the tree supporting the leaf indefinitely, theoretically. But they don't do that because they're like it's kind of like with your hair, right? Like you could the hair inside the follicle, right, is still alive and supported. Once that dies, it falls off and starts again. Some people like your eyebrows, right? That time's very short, and so you don't have like super long eyebrows, right? Because it's just short. Hair grows longer. Theoretically, you can have it forever, but you don't. Trees don't do that stuff. I'm done supporting the trees for like months now. Yeah.
Good, my time.
Yeah. But basically like a pine tree, right? Like, yeah. Sweet. That was a good question.
I learned about these. What is the role of pigments?
Yeah.
They're not needed. They make the plant look pretty. Because I know that they can take in purple leaves and oranges and reds, but not really like green and yellowish, but oh,
That's what it is. Besides the that gets reflected.
gotcha.
Okay, I realized it wasn't tons of time, but we're going to talk about things general. How is it going? Your buddy's doing okay? Cool. I've heard several good answers. This is good. It's harder to explain. Even if you think you know, it's harder to explain, which is why it's good for you. Help me out here. We're not going to go through all of them, but just help me out as a group. So, anybody have small children in their lives? Nieces, nephews, cousins, kids? Okay. Your small relative or neighbor or friend or whomever asks you, why is grass green? The lame answer is because it's pretty. I mean, it's nice, but like, because it's pretty, right? No. You are going to answer, why is grass green? Why is grass green? You ready to launch into this long-winded explanation and blow their little minds? Right? No, it's not how you... Briefly, this is not relevant. Briefly, as a child, I didn't like going to bed at night, which, I mean, I still don't. But my sister is a couple years older than me. She's like 2 years older than me. Right before bed, she'd come up with some science question to ask my dad, and he'd leap into heroic action to, like, draw all the little things. And my mom would be like, they're playing you. He's like, no, they want to learn. We were playing him, but, you know, anyways, it was cool. So, okay, but you're explaining to some child who wants to get out of going to bed, why is grass green? Why is grass green? Yeah, help us out.
The green pigment's reflected, green light's
Yeah.
reflected.
Yeah, beautiful. Okay, so Dax is right, right? Green light's reflected, this pigment's caught this light. Okay, now what? This kid's trying really hard to not go to bed. So they're like, well, what happens next, right? Because they don't know what's going on, but next thing. So the pigment's absorbed a lot of light. Not the green stuff, but everything else. It's absorbed the green light. Cool. Now what's going on?
The light is used to power electrons
Yeah.
that make ATP.
Yeah, perfect. Are you cool with it? That light under that light under the catchment pigments is going to power stuff. It's going to power its electrons. It's going to make ATP. It's going to then go make sugar. We're cool with that? Great. Okay. One last sneaky question, then I'll let you go. I promise I'll let you go. One last sneaky question. So, our friend the plants give us oxygen, right? Cool. Where are our friends the plants getting the oxygen? They can't just like poof make it from nothing. So where do they get the oxygen from that they then give off for us? Water, right? Are we okay with this? Sorry, too many things. Here, right? That's the oxygen they're taking it from water, giving off for us. We're good? Sweet. That's all we've got. I'll see you on Friday. Friday is going to be a catch-up and review day. Do come if you can. If you can't, like, do come. It's going to be helpful, I think. You do have a quiz on Friday, though.